Process of formation of the megaspores from the megaspore mother cell, it is called as Megasporogenesis. Egg apparatus consists of 2 synergids and 1 egg cell. Hello everyone, a warm welcome to today's session on sexual reproduction in flowering plants. I am Dr. Divya, faculty in biology, Vidhyaashram Pre-University College, Mysuru, a temple of excellence. So to begin with today's session, let us brush what we have learned in the previous sessions. So in the previous session, we had learnt about the pre-fertilization structures. So in the process of reproduction in a flowering plant, if the pre-fertilization events has to occur, so what is the pre-fertilization events? Those events that occur before the fertilization, it is nothing but the pre-fertilization events. So if the pre-fertilization events has to take place in a flowering plant in terms of sexual reproduction, then various structures are involved. So what were the structures that we had studied? We had studied about the male reproductive structure, that is the anther and the filament which constitutes the stamen. So inside the stamen or inside the anther, I told you there is the microsporangium, and inside the microsporangium we had studied about the microspores or the pollen grains. And when it comes to the female reproductive structure that are involved in the pre-fertilization events, we had studied about the gynoecium or the pistil. So in the gynoecium or the pistil, you have the ovary, and inside the ovary you have the ovule, and inside the ovule you have the embryo sac. So embryo sac plays a major role in bringing about the pre-fertilization event. So that is why we had also learnt about the structure of the embryo sac. So moving further to today's session, we will discuss what are the different pre-fertilization events that occur in a flower in terms of sexual reproduction. So there are 2 main events that occurs. One is the microsporogenesis and the other one is the megasporogenesis. So we'll study about them one by one. So obviously we know that microspores, it is nothing but the pollen grains. So where are the pollen grains produced? They are produced inside the microsporangium. So where is the microsporangium present? It is present in the anther, right? So what is an anther? It is the male reproductive part of a flower. So therefore, microsporogenesis takes place in the male reproductive part of the flower. Then we'll study about the megasporogenesis. So what is this megasporogenesis? So where are the megaspores present? They are present in the ovary of the female gametophyte. So in the ovary, what you have? The ovules. In the ovules, what do you have? You have the embryo sac. That is why whatever events that occur inside the embryo sac, we call it as the megasporogenesis. So let us briefly discuss about these 2 pre-fertilization events, that is the microsporogenesis and the megasporogenesis. So first we'll learn about the first reproductive event, that is the microsporogenesis, which comes under the pre-fertilization events. So microsporogenesis, as I told you, it is the process wherein the microspores. So what are the microspores? The microspores are nothing but the pollen grains. So where are these pollen grains produced? They are produced in the pollen mother cell. So where is the pollen mother cell present? The pollen mother cell is nothing but the sporogenous tissue. So what we'll learn here is, again I will teach you the diagram of the microsporangium so that it will be even more clearer. So I'll draw a diagram of the microsporangium. So you know that the microsporangium, it is tetragonal. It has 4 sides. So what is there in the microsporangium? You have different layers that cover the microsporangium. So you have in the microsporangium, you have the epidermis. One of the first layer that covers the microsporangium is the epidermis. It is the outer layer. So you have the epidermis. So epidermis. So epi means outside. Dermis, as I told you, it is derma, means the skin or the covering. So epidermis. Below the epidermis, you have the endothecium. Endo means inside. So you have the endothecium. So this is the endothecium. So inner to the endothecium, you have the middle layers. So these are the middle layers. Middle layers. And inside the middle layer, there is a very important layer called as tapetum. So where is the tapetum present? It is the layer that is present around the pollen sac. So this layer is the tapetum. So what is the function of the tapetum? So whatever pollen grains are developing or maturing inside the pollen sac, those pollen grains get the nourishment from the tapetum. So therefore, tapetum is the layer which acts as a nourishment, or it acts as a layer that provides nutrition or nourishment to the developing pollen grains that are present inside the pollen sac. So here you have, inside this, you have the microspores. So before that, you will have the sporogenous tissue. So what is a tissue? Tissue is a group of cells. You'll have the sporogenous tissue. So this is the sporogenous tissue. So you have the sporogenous tissue. So why is it called a tissue? Because a group of cells are involved in this. That is why it is called as a tissue. So one cell, you call it as the pollen mother cell or the microspore mother cell. You can call it as the pollen mother cell or the microspore mother cell. So this pollen mother cell or the microspore mother cell is the one which develops into the pollen grain. So that is what I have written there. The process of formation of microspores from the pollen mother cell through the process called meiosis. So here meiosis will occur. Meiosis is nothing but the reduction division will take place. So once the reduction division of the pollen mother cell takes place, the pollen grain or the microspores are being produced. So that is nothing but microsporogenesis. Why you call it as microsporogenesis? Because it takes place in the microsporangium, which is a part of the anther, which is the male reproductive structure of the flower, and this microsporangium produces the microspores. So microsporogenesis. Microspores are the minute spores. They are also called as the pollen grains. Genesis is to generate something or to develop something new, that is called as genesis. So what is taking place here? Here in this particular process, the microspores are being generated or developed from the sporogenous tissues or the sporogenous cells that are present inside the microspores. So therefore, the process is called as microsporogenesis. So we'll study now how the steps are involved, or what are the steps that are involved in microsporogenesis. So in microsporogenesis, you know that when the anther develops, the cells of the sporogenous tissue, they undergo meiotic division. That is what I have shown you here. So when an anther develops, you take a cross section of the anther, wherein that is cross section or the transverse section of the anther, wherein you see the microsporangium. So when you look at the microsporangium under the microscope, you can find that it has the microspore mother cell or the pollen mother cell. So that pollen mother cell or the sporogenous tissue or the sporogenous cell in general, they will undergo meiotic division, which is a reduction division to produce or to form microspore tetrads or the pollen tetrads. So you have here the microspore. You have the microspore. So where are these microspores present? They are present here inside this, inside the pollen sac, or in the pollen mother cell, inside the pollen sac. So this particular microspore, they undergo meiosis. So they undergo meiosis and they produce the pollen tetrad. So this is the pollen tetrad. So first they undergo first round of meiotic division, and again they undergo second round of meiotic division. And they form the microspore tetrad or the pollen tetrad. Why is it called a tetrad? Because 4 pollen grains are there inside this. 4. That is why it is tetra. Tetra means 4. That is why it is called as a tetrad. So this is how the microspores develops inside the microsporangium through a process called as microsporogenesis. So next, moving further, there are again certain steps involved inside the microsporangium, that is in the pre-fertilization events in the male gametophyte, which is once the microspore tetrad is formed, again certain steps are involved wherein the microspore tetrad becomes separate pollen grains, and what occurs inside the pollen grain? The pollen grain has to mature. How the maturation and will occur, we'll study. So each cell of the sporogenous tissue will give rise to a microspore tetrad. So this is the microspore tetrad that I was talking about. So each cell of the sporogenous tissue will give rise to a microspore tetrad. So once the microspore tetrads are formed, the anthers will mature. So you know that the size of the pollen grain inside will increase. Whatever vegetative cell or the generative cell that has to be formed, every process will take place inside the microsporangium. So when there is no space, and also the anthers, they will get desiccated or they will become dehydrated. So once dehydration occurs, what happens? The anther wall will become brittle or it will become hard. So when favorable conditions occur, they will just burst open and the pollen grains will dehisce out or it will come out of the anther. So that is what happens. So here, once the microspore tetrads are formed, the anthers are called as mature anthers. So it is time for the anthers to burst open and liberate the microspores or the pollen grains. At that time, what happens? The microspores will dissociate from each other. So why dissociating from each other? Because first inside the anther, it will be in the form of a pollen tetrad having 4 pollen grains together like this. So these pollen grains should separate, they should dissociate. So once they dissociate, the anthers will burst open. So when they dissociate, what happens is, for one microspore, when they dissociate, 4 microspores will result. So there is no space inside the anther. The anthers will burst open and the pollen grains will start to come out of the anther. So they will dissociate from each other and form the pollen grains. So once the pollen grains are formed, the anthers will dehisce. Dehisce is nothing but splitting open of the anther. Remember, I told you, when you observe an anther, it is made up of 2 lobes. So this is the filament of the anther. When you observe the anther, it is made up of 2 lobes. So hence it is bi-lobed. So I told you there is a groove. There is a small split here, a small groove here. And this groove, actually, it will result in splitting open of the anther. So when the anthers, they split open in this particular groove, when they split open, the pollen grains will start to come out. They will start to come out. Some of the pollen grains may fall to the ground. Some of the pollen grains may just remain on the anther. Some of them may be they'll just move away. If the pollen grains are very light, they'll be carried by the wind. So that you'll study in pollination in the next coming session. So this is what happens in the case of Microsporogenesis. So now the anthers have burst open, so and the pollen grains have come out. So again I'll explain you what is there in the pollen grain. Inside a mature pollen grain, you will have the vegetative cell. So you have learnt about this in the previous session. You'll have the vegetative cell and you'll have the generative cell. Generative cell. So this vegetative cell, it actually provides nourishment to the generative cell. Apart from that, the vegetative cell has no other function. It is the generative cells that develops further into the two male gametes and they enter into the ovary of the female gametophyte. But the vegetative cell has no other function in reproduction, but their only function as of now is to provide nourishment or nutrition to the two gametes that are formed. So this is about the pre-fertilization event that occurs inside the microsporogenesis, which is the male gametophyte. So next we'll study about megasporogenesis. So microsporogenesis, it was related to the stamen, that is the anther. Megasporogenesis, it is related to the female gametophyte, that is the gynoecium. So in megasporogenesis, so we'll study about the next pre-fertilization event. So whatever pre-fertilization event had to occur inside the anther, it is over. We have learnt about it. So now we'll move on to what are the pre-fertilization events that will occur inside the female gametophyte, in this case in the megaspore. So the process of formation of megaspores from the megaspore mother cell, remember in the case of microsporogenesis, it was the process of formation of the microspores or the pollen grains in the microspore mother cell or the pollen mother cell. It is called as microsporogenesis. But in the case of megasporogenesis, I am talking about the female gametophyte, the events, the pre-fertilization events occurring in the female gametophyte. So in this case, in the case of megasporogenesis, the process of formation of the megaspores from the megaspore mother cell, it is called as megasporogenesis. So we'll study about what are the steps that are involved in megasporogenesis. So the megaspore mother cell, it will, before forming a complete embryo sac, so at the end, the embryo sac is the one that has to be formed if the fertilization process has to continue. So here, first we'll study how the megaspore mother cell will develop into a embryo sac. So the megaspore mother cell, it will undergo meiotic division to produce 4 megaspores. So as you can see here, I can show it to you in a different diagrammatic representation. So you have the megaspore mother cell having one nucleus, one cell, one nucleus. So this will undergo meiosis. So this will undergo meiosis, that is meiotic division will take place. It will undergo meiosis to produce two cells. The same thing you can observe here. One cell, which is the megaspore mother cell, this cell will undergo meiosis or reduction division to produce 2 cells, and again it will undergo one more meiotic division to produce 4 cells having its own nucleus. So what happens here? The megaspore mother cell, it undergoes meiotic division to produce 4 megaspores. So out of the 4 megaspores, can you see here? Megaspore 1, 2, 3 and 4. So out of the 4 megaspores, these 3 will get degenerated. So they will get degenerated and only 1 will be functional. So the other 3 will just get degenerated. The other 3 will get degenerated and only the 1 will be functional. So this functional megaspore is the one which develops into the embryo sac. So this functional megaspore is the one which develops into the embryo sac and the 3 will degenerate. So this is how the megasporogenesis occurs. So I'll explain once again. So you have the megaspore mother cell which undergoes the reduction division called as meiosis to produce 2 cells. The 2 cells again undergoes meiotic division to produce 4 cells. So out of the 4 megaspore mother cells, out of the 4 megaspores, the 3 megaspores will degenerate. They are of no use. Only 1 will be retained, and only that 1 megaspore that is retained, it will produce the embryo sac or the megaspore. It will produce the embryo sac. So only this is the functional. So only this is the functional megaspore. Only this one is functional. It is the functional megaspore. So next, again, the functional megaspore is the one I told you. It develops into the female gametophyte. So the functional megaspore is the one which develops into the female gametophyte. It is called as the embryo sac. So how this embryo sac develops, we'll study briefly again. So there is one more term which you have to remember here. It is called as monosporic development. Mono means one. So the method of formation of embryo sac from a single megaspore is called as monosporic development, right? This is a monosporic development. Why? 4 megaspores were formed. Out of the 4 megaspores, 3 got degenerated. Only 1 was left behind, and only that one which was left behind will develop into a embryo sac, right? So that is why it is called as monosporic development. So the monosporic development is the method of formation of embryo sac. It is a method of formation of embryo sac. Remember, formation of embryo sac from a single megaspore. So it is called as monosporic development. So this was about the formation of the embryo sac. So again, now the embryo sac, I told you, embryo sac will be formed. So how will that one single megaspore that was retained, how it will develop into the embryo sac, we'll just look into. So again, you had one cell was retained, right? Because of monosporic development. Why? Though there were 4 megaspores, the 3 got degenerated or it was lost. It is of no use. Only the one that is remaining will develop into the embryo sac. So here what happens is the functional megaspore will develop into the female gametophyte or the embryo sac. So what are the steps involved in the development of the embryo sac? We are going to study. So the nucleus of the functional megaspore. So which is the functional megaspore? This is the functional megaspore having the nucleus. It is monosporic. It is only one cell, hence it is monosporic. So this functional megaspore having the nucleus of the functional megaspore will divide. So remember, here cell will not divide. The nucleus will divide. So there is increase in the chromosome number. When the nucleus divides, obviously the genetic material also divides with it. So here there is increase in the chromosome number wherein it will divide. That is why, since there is the chromosomes number will double, right? Hence it is considered to be mitotic division. So here what happens is this particular megaspore that was retained, it will undergo mitotic division. So it will undergo mitotic division, mitosis. What? Not the cell, but the nucleus will undergo mitotic division and 2 nucleus will be formed. Again, a second round of mitotic division will take place and 4 nucleus will get formed. Again, one more third round of mitotic division will take place and you'll have double the amount of nucleus. Now here 4 nucleus were there, now you have 8 nucleus. So this is how the division takes place. So what are the steps that are involved in the formation of embryo sac? The nucleus which is present in the functional. So always write it as functional megaspore. Why it is functional? Because only the functional megaspore is the one which develops into the embryo sac. The rest of the 3 is of no use. It is non-functional. They will get degenerated. So the nucleus of the functional megaspore, they will undergo mitosis or mitotic division and they will form 2 nuclei. The 2 nuclei later on, it will move to the opposite poles. Can you see here? It is moved to the opposite poles. Why has it moved to the opposite poles? Remember, in the 2 poles of the ovule, you had the antipodal cells and the egg cells and the synergids, right? So, if the cell has to form, that is why the nucleus divides to 2 poles. Again, this nucleus will undergo mitotic division and it will form 2 nucleus at one end and the other 2 nucleus at the other end. Again, mitotic division will take place and 4 nucleus will be formed at one end and the 4 nucleus at the other end. So, the 2 nuclei will move to the, it will move to the opposite poles and it will form the 2 nucleate embryo sac. So, now this is the 2 nucleate embryo sac. So, this is a 2 nucleate embryo sac. So, after the formation of the 2 nucleate embryo sac, 4 nucleate embryo sac is formed. How? Again, the 2 nucleate embryo sac will undergo mitotic division to form 4 nucleate embryo sac. Now, this is 4 nucleate. So, you have the nucleus here. Here also, you'll have the nucleus. And again, so the 2 nuclei again divides mitotically to form 4 nucleate. The 4 nucleate again divides mitotically to form the 8 nucleate embryo sac. So, now this is the 8. Again, this will undergo mitotic division to form the 8 nucleate embryo sac. 8 nucleate embryo sac. So, after the 8 nucleate stage, the cell walls will start to develop. So, once the cell wall starts to develop around this nucleus, so around this nucleus you have the cell walls being developed. Then only it will be cell, called a cell. So, remember I told you in the previous session, the embryo sac, it is a 8-celled, right? It is 8-celled. So, you have now, once the cell wall develops around the nucleus, now the embryo sac has 8 cells. 1, 2, 3, 4, 5, 6, 7 and 8. It has 8 cells. So, out of the 8 nuclei, 6 will get surrounded by the cell walls and they get organized into the cells. So, why 6 will get surrounded by the cell walls? Because the other 2 will form the central cell, right? The other 2 will form the central cell. So, inside the central cell, you'll have 2 nuclei. That is why the 6, 1, 2, 3, 4, 5, 6 or 1, 2, 3, 4, 5, 6, anything. So, only 6 will form the cell wall. 6 nucleus separately they'll form the cell wall and the 2 nuclei they'll move to the center and around the 2 nuclei one cell wall is formed. Therefore, it will become 7-celled. So, that is how it occurs. Though it is 8 nuclei, it will become 7-celled. So, the embryo sac, first it will be 8 nucleate, but once the cell wall surrounds the 8 nucleus, or we can say it as once the cell wall surrounds the 6 nucleus and the 2 nucleus will move to the remaining 2 nucleus will move to the center and it will get surrounded by one more cell wall. It will form a 7-celled embryo sac. But how many nucleus are there? 8 nucleus are there. But how many cells are there? 7 cells are there. So, this is how the embryo sac development takes place. So, the remaining 2 nuclei, as I told you, the remaining 2 nuclei will move to the center and they are called as the polar nuclei. So the remaining 2 nuclei, it will move to the center and you can call it as the polar nuclei. So you can see here cell walls are developed. So now you have 1 cell, 2, third cell, this is the 4th, 5th cell, and this is the 6th cell. So remaining 8 nucleus, right? Only 6 cells having 1 nucleus each is formed. What about the other 2 nucleus? It will move towards the center and a cell wall will be formed around it. So this is the central cell. It will become the 7th cell. So this will be the 7th cell. So this is how the embryo sac development takes place. So after that, what happens is, as I told you, the remaining 2 nuclei, it will move to the center and they are called as the polar nuclei. They have come from the 2 poles, they have moved to the center and now they are called as the polar nuclei. So the 3 cells, whatever is there at the micropylar end, so you have the micropylar end. Always remember, don't get confused. When you look at a ovule under the microscope, whichever a region you find an opening, right, that will be the micropylar end. Though it is inverted, or sometimes the diagrams may look like the micropylar end is towards this side, or sometimes it will be towards this side. It always depends on where the opening is there. So what is that particular opening? It is the micropylar opening. Why is there an opening? Because I told you the ovule is surrounded by integuments, right? But there is a region in the ovule where integument is not covering the ovule. That particular region, there is a gap there. So that is the micropylar region. So here you have the micropylar region or the micropylar end. Then you have the chalazal end. Chalazal end. So what happens here is 3 cells will get grouped together. 3 cells will get grouped together at the micropylar end and they are called as, or they constitute the egg apparatus and 2 synergids. So 2 plus 1, 3. So egg apparatus. So together it is called the 3 cells that are at the micropylar end. Together they are called as the egg apparatus. So what is there in the egg apparatus? Egg apparatus consists of 2 synergids and 1 egg cell. So here you can see, so this overall together you can call this as the egg apparatus. So what is there in the egg apparatus? You have 2 synergids, and you have 1 egg cell. So this is the 3 cells that were present in the micropylar end. They developed into the egg apparatus. So in the egg apparatus, you have 2 synergids and 1 egg cell. And the other 3 cells that got grouped together in the chalazal end, you call it as the antipodal cells. So you have the antipodal cells. So these 3 cells at the chalazal end, you can call it as antipodal cells. Antipodal cells. So the remaining 2 cells which is in the center, it is the central cell. So this is how the embryo sac develops. So first the embryo sac, it was uninucleate. The nucleus underwent mitotic division to form 2 nucleus, again mitotic division to form 4 nucleus. Out of the 4, again mitotic division to form 8 nucleus, and once 8 nucleus got formed, 6 nucleus got surrounded separately by the cell wall and 2 nucleus got surrounded together with the cell wall, and you had 7 cells. So which were the 7 cells that were formed? 3 cells at the micropylar end called as the egg apparatus, which consists of 2 synergids and 1 egg cell, and the other 3 cells at the chalazal end called as the antipodal cells, and one more cell at the center. In the central cell, there are 2 nucleus. That is why an embryo sac, it is 8 nucleate and it is 7 celled. So this is about the pre-fertilization event that occurs in the female gametophyte, that is in the gynecium or the pistil, especially inside the ovule, wherein the embryo sac will get developed. So this was about today's session. So in the next coming session, we will study about the pollination, the kinds of pollination and the agents of pollination, because once the pollen grains or once microsporogenesis is developed and once megasporogenesis takes place, the next step is fertilization, wherein double fertilization will occur. If the double fertilization has to occur, then whatever pollen grains that developed in the anther, right? I had told you whatever pollen grains had dehisced or come out of the anther, that should be pollinated. And how pollination will occur? By either by wind, by insects, by water, etc. So therefore, pollination is the very important event. So that pollen grains will be carried to the stigma of the same flower, or it can be carried to the stigma of the different flower of the same species. And once the pollen grains get deposited in the stigma, it will germinate and it will carry the 2 male gametes through the pollen tube into the ovary. So if you need to learn about all that, that is the double fertilization event, first you need to know about the pollination. So in the next coming session, we will focus on pollination, what are the different types of pollination and what are the different agents that help in the carrying of the pollen grains from the anther to the stigma, or the agents of pollination. So I hope you have understood this session very well. So in the next session, we shall meet again, and thank you.